Here are Puerto Rico Medical Board-style clinical vignettes focused on apoptosis, with 4 answer choices and detailed explanations. Clinical Vignette 1. Intrinsic Pathway. A 62-year-old man with advanced prostate cancer is started on chemotherapy. After several days, tumor cells begin to shrink due to activation of mitochondrial cytochrome C release and caspase activation. Which of the following best describes the process involved? A. Necrosis. B. Autophagy. C. Apoptosis. D. Pyroptosis. Apoptosis. Explanation. Apoptosis is a programmed cell death mechanism often activated in response to DNA damage, hypoxia, or cytotoxic drugs. The intrinsic mitochondrial pathway involves the release of cytochrome C, which activates caspase 9, leading to a cascade that results in controlled cell death without inflammation. Clinical Vignette 2. BCL2 function. A researcher blocks the function of the BCL2 protein in cultured cells. As a result, the cells undergo rapid mitochondrial membrane permeabilization. What is the likely consequence? A. Cellular hypertrophy. B. Inhibition of cell cycle. C. Enhanced survival. D. Apoptosis. D. Apoptosis. Explanation. BCL2 is an anti-apoptotic protein that stabilizes the mitochondrial membrane. Blocking it promotes the release of cytochrome C, leading to activation of caspases and apoptosis. Clinical Vignette 3. Morphological features. A liver biopsy from a patient with hepatitis shows hepatocytes with eosinophilic cytoplasm, cell shrinkage, and nuclear fragmentation without signs of inflammation. What process is most likely occurring? A. Casus necrosis. B. Liquefactive necrosis. C. Apoptosis. D. Fat necrosis. Answer C. Apoptosis. Explanation. Apoptotic cells show cell shrinkage, chromatin condensation, eosinophilic cytoplasm, and formation of apoptotic bodies, all without inflammation, distinguishing it from necrosis. Clinical Vignette 4. Death receptors. A tumor cell expresses high levels of Fas ligand, FASL, on its surface. It binds to Fas CD95 receptors on T lymphocytes, inducing cell death. What type of apoptosis pathway is activated? A. Intrinsic. B. Caspase-independent. C. Necroptosis. D. Extrinsic. Answer D. Explanation. The extrinsic pathway of apoptosis is triggered by death receptors like Fas, CD95, or TNF receptor. These receptors activate caspase 8, leading to cell death via the apoptotic pathway. Clinical Vignette 5. Apoptosis versus necrosis. A patient experiences ischemia to the kidney after a hypotensive episode. After 4 hours, histological exam shows swelling and inflammation. After 24 hours, some cells are shrunken with intact membranes and no surrounding inflammatory response. What does this indicate? A. Necrosis occurred first, followed by apoptosis. B. Only necrosis occurred. C. Only apoptosis occurred. D. Autophagy occurred. Answer. A. Necrosis occurred first, followed by apoptosis. Explanation. Acute ischemia leads to necrosis due to ATP depletion. However, some cells may undergo apoptosis later, especially in partially injured areas, leading to cell shrinkage and no inflammation. Clinical Vignette 1. HER2-neu. A 45-year-old woman presents with a rapidly growing breast mass. Biopsy confirms invasive ductal carcinoma. Immunohistochemistry reveals overexpression of a tyrosine kinase receptor involved in cell proliferation. Which gene is most likely amplified? A. BRCA1b, TP53C, HER2-neu, ERBB2, DRB1, ERBB2. Explanation. Explanation-neu is an oncogene encoding a tyrosine kinase receptor. Its overexpression leads to uncontrolled cell growth and is seen in 20 to 30% of breast cancers. Unlike tumor suppressors like BRCA1 or RB1, oncogenes are activated by mutation or amplification. Clinical Vignette 2. RAS mutation. A 58-year-old man with colorectal cancer is found to have a mutation in a gene involved in GTP binding signal transduction. Which of the following is the most likely gene affected? A. APC. B. RAS. C. BCR-ABL. MYC. Explanation. RAS is a proto-oncogene encoding a GTP haze. Mutations result in constitutively active RAS, leading to unregulated proliferation. It's one of the most common oncogene mutations in human cancers, including colon, pancreatic, and lung. Clinical Vignette 3. BCR-ABL. A 34-year-old man presents with fatigue and splenomegaly. Lab tests reveal markedly elevated WBC count. Bone marrow biopsy shows increased granulocytes. A T92 chromosomal translocation is found. Which oncogene is involved? AMIC, BCL2, BCR-ABL, D, RAS. Answer C. Explanation: the Philadelphia chromosome is a T922 translocation creating the BCR-ABL fusion gene, which encodes a constitutively active tyrosine kinase. It is seen in chronic myelogenous leukemia, CML, and treated with imatinib. Clinical Vignette 4, Mice Overexpression. A child presents with a large, rapidly growing abdominal mass. Biopsy reveals a starry sky pattern. Cytogenetic studies show a T814 translocation. Which gene is involved? A, NMIC, B, BCL2, C, CMIC, D, TP53. Answer C, CMIC. Explanation: Burkitt lymphoma is associated with CMIC activation due to T814, which places the gene under the control of the Ig heavy chain enhancer on chromosome 14. This drives rapid B-cell proliferation. Clinical Vignette 5, NMIC in neuroblastoma. A 3-year-old child presents with abdominal distension and a firm mass crossing the midline. Urine is positive for elevated HVA and VMA. Biopsy confirms neuroblastoma. Which gene is likely amplified? A, NMIC, B, RETS, C, KRAS, D, BRCA2. Explanation: NMIC amplification is a poor prognostic factor in neuroblastoma, a tumor of neural crest origin. It often produces catecholamine metabolites, HVA-VMA. NMIC is a transcription factor oncogene. Would you